According to Alzheimer's.org, BMC Geriatrics, and the National Institute on Aging, around 40 to 65% of the people with Alzheimer's disease have the APOE4 gene. The APOE4 gene increases risk, and if you have two copies, one from each parent, you are at an even higher risk. This gene may make the symptoms of Alzheimer's disease appear at a younger age. The risk of this gene differs based on populations. In America, about 20 to 30% have the APOE4 gene, and only about 2% have two copies. These genes are estimated to account for 1% or less of Alzheimer's cases. Overall, about 70% of the cause of Alzheimer's disease is due to genetics. This leaves the other 30% to environmental factors. Some of these include air pollution exposure, heavy metals, pesticides, industrial chemicals, electromagnetic fields, and vitamin D deficiency. An example of an altered protein in people with this trait is the beta amyloid protein.
23andMe tests the epsilon 4 variant in the APOE gene. This gene is involved in making apolipoprotein E, which is a protein that helps carry cholesterol and other types of fat in the bloodstream. This is an important process that is thought by scientists to contribute to the development of Alzheimer's disease when it encounters issues. The APOE gene comes in several forms, called alleles, epsilon 2, epsilon 3, and epsilon 4. While the first two are not tested by 23andMe, APOE epsilon 4 is known to increase risk for Alzheimer's and is associated with an earlier age of disease onset. About 15% to 25% of people have this allele, and 2% to 5% carry two copies, which is more dangerous than having only one. Despite inheriting epsilon 4 increases a person's risk of Alzheimer's, some people with this variation never experience symptoms, and many who do develop the disease do not have any copies of it. Also, the determinist genes APP, PSEN1, and PSEN2 have variations in their DNA where, if a person carries one, chances that they will develop Alzheimer's dementia are higher. The 23andMe test does not look for these gene mutations.
The two genes that our test would include as SNPs that are not tested by 23andMe are the APP and PLD3 genes. Both genes have crucial roles in the development of Alzheimer's disease in individuals. The RS identifier of the APP SNP is RS63750847, and the different versions include AA, AG, and GG. For the genotype AA, the MAG is 4, and the combination results in resistance to Alzheimer's disease. For AG, the MAG is 3.5, and the different alleles make the person a carrier for reduced risk of Alzheimer's disease. The final allele combination is GG, which results in a normal risk for the development of the disease with a MAG of 0. This means that there is no resistance for the development of Alzheimer's. Those who are at risk develop a buildup of the amyloid protein, which is thought to hinder nerve cell function in the brain. The RS identifier of the PLD3 gene is RS145999145 and has 3 versions, AA, AG, and GG. AA and AG versions double the risk for Alzheimer's disease, while the GG version doesn't decrease or increase the risk. These different versions lead to changes in the PLD3 gene, whose normal functions are regulating membrane composition and functions, which helps with processing and trafficking proteins within neurons. This maintains cellular homeostasis by recycling essential components, and it is believed to have a role in amyloid precursor protein processing.
Genetic tests like 23andMe have notable limitations. While they test for APOE Epulsion 4LI associated with Alzheimer's, they miss crucial genes such as APP, PSEN1, and PSN2. This deterministic gene significantly raise the risk of early onset of Alzheimer's, but 23andMe does not include them in their testing. Furthermore, carrying APOE Epulsion 4 only increases risk, it does not guarantee the disease. This uncertainty often leads to confusion as many people with ELI never develop Alzheimer's, while some who don't have it still get the disease. So many genetic variations are still unknown. So the test may miss other factors contributing to the disease. Finally, Environmental influences that are diet, lifestyle, and health are crucial in determining Alzheimer's risk, which genetic tests can't predict. Ethically, genetic testing for Alzheimer's possesses several challenges. Receiving a positive result for a risk of ALI can cause emotional distress, anxiety, or depression, as Alzheimer's has no cure. The financial risks are also significant associated with it, as with the potential genetic discrimination in the life insurance. This, despite protection like GINA, furthermore, there's a need for clear, informed consent. Consumers might misunderstand the risk associated with the results, leading to an unnecessary fear or false reassurance. Lastly, privacy concerns arise with the handling of sensitive genetic data, especially if companies share it with third parties without the customer's explicit knowledge.
According to the article by McConnell, emotional distress is not the only ethical consideration. Financially, those who become diagnosed must bear the burden of significant increases in healthcare costs in order to monitor and help treat the disease. Even with insurance, it is not guaranteed that these costs will be completely covered, leaving families to struggle to pay for treatment. Becoming aware that one may develop Alzheimer's disease could also affect their employment. If they begin to experience symptoms and inform their bosses, it is likely that they could be discriminated against and fired for something completely out of their control. Based on these points, it is evident that genetic testing for Alzheimer's disease can have significant emotional and financial impacts on affected individuals.